Okay, today we're going to be doing a demonstration on mitosis. We've already studied mitosis in class, and we are going to be doing a review, a very simplified demonstration to show you mitosis. So the first thing we're going to start with is our cell. And in our cell, we have our nucleus. There we go. And within our nucleus, we have our DNA, which is uncondensed in its uncondensed form. So when you first start looking at the cell cycle, the first we want to look at interphase, which is gap 1, S1, and gap 2, or G2. So in G1, what happens is the cell actually lays down or produces more organelles and grows in size. In the next phase, S phase, is our synthesis phase when our DNA duplicates. Bam! We got double the DNA. And we're ready to move on to the next gap phase, where the cell continues to grow and produce its organelles and gets ready for mitosis, or cellular division. So let's move on to our first, which is prophase. In prophase, what happens is our nuclear envelope disintegrates, our DNA becomes condensed. Let me condense the DNA for you. And we started off with 2 chromosomes, and now what you have is still 2 chromosomes, but in its duplicated form, they're still each called a chromosome, but they have 2 parts, which is their chromatid. So each chromosome is made up of 2 chromatids, and they are now in their condensed form, and they are attached via their centromeres or kinetochore, and that is being duplicated by our little mini clothespins. So now we have no nuclear envelope. Our centromeres also duplicate in prophase, and the one starts to migrate to the opposite pole. And you know you are at the end of prophase when you have condensed chromosomes and 2 centrioles at opposite poles. The next phase is metaphase, and what happens is our centromeres start laying down the cytoskeleton our microtubules that attach via the kinetochore to each chromosome, and they line up along the metaphyseal plate. So when you're looking at a slide and you see your chromosomes all lined up along the middle of the cell, you know you are in metaphase. Our next phase is anaphase, and what happens is these kinetochores connected to the chromatid start pulling the chromosomes to opposite ends of the pole via those centrioles and the microtubules. When you have your condensed chromosomes at each end of the cell, you are at the end of anaphase. The other thing that starts to happen in anaphase is you might see a small change in the shape of the cell, because cytokinesis is not really distinct as to when it starts and when it ends. It is by the time cytokinesis, or the separation of the two cells, is complete, mitosis is complete. But the changing in the cell size can start at the very end of anaphase or at the beginning of telophase. Speaking of telophase, we are now at our last phase of mitosis, and what happens is the cell replaces the nuclear envelope around both sets of chromosomes, and then cytokinesis starts pinching or separating the two cells so that at the end of your cell cycle, you have two cells with an exact duplicate copy of chromosomes.